Hello, Michael and Rachel. Thank you for taking the time to join this discussion. Would you like to introduce yourself and tell us about your field of research?
Hi, I'm Rahel. I'm a PhD student at the University of Zurich at Michael Bautis's research group. And what I'm focusing on in my project is cancer cell lines and their variants and their possible and how similar they are to their origins.
Mm-hmm. Thank you. And can you tell me something more about the data that you are currently possess?
So in the database for our group, we have cancer copy number data. So we have over 140,000 variants. But as my project focuses on cell lines only, and we collect the cell line variant data, so both copy number and single nucleotide variants, and as well as additional annotations to that. And it's also very important for us to collect known publications about cell lines, cancer cell lines.
Okay, thank you, Rachel. And Michael, please go ahead, introduce yourself. And I'd like you to tell me about, with regards to the INODE's nature of enriching and combining various data sources. What is your motivation of leveraging the INODE ecosystem?
Yes, so thanks for the question. So just as a brief introduction, I'm a professor of bioinformatics here at the University of Zurich, and our main area of research is in cancer genomic variation, as Rahel already said, and we focus on copy number variations or structural genome aberrations in tumor samples and on the different patterns between different diseases, genes stay involved, what possible functional mechanisms could be derived from that. And for that, we have a large database, as Rahel already said, with around 140,000 genome profiles for these structural aberrations and additional data like the cell line data, which is more of a focused project. So in regard for these copy number variants, we are best of class with our resources. It's also completely open, everybody can use it and so on. However, it's pretty shallow in its information content. So we have a lot of information about these genomic variations, obviously this complete genome profiling data, but then we have also a little bit of additional data about the samples, the diagnosis, everything curated, technical, provenance, and so on. This is all regarding with these samples. However, this resource becomes more useful if you then, when identifying some region of interest, for instance, in a certain tumor type, where you see certain genomic variations being recorded and shown in our database, then to make the jump there to get additional information, about functional annotations, about which genes are there, about literature, which leads you to explore more knowledge instead of only data. So we have lots of data, but the INODE, the combination of INODE now helps us to make more of a knowledge discovery from the data we have. So that using our data as a jumping point to then really find new ideas about what could go on in the cancer physiology and so on of the samples we have annotated. So the knowledge part is what is being brought in or greatly improved by having a system as INODE connected to our data. And we start this by focusing on cell lines where we already have additional information curated during our code.
Developed by INODE and the Bautis Research Group from the University of Zurich and aims to help biomedical researchers in identifying strong or discarding weak correlations between cancer types and the genome. The application is composed by two tabs. The first tab provides copy number variation plots and a relative cytogenetic band information that were obtained from the Progenetics API. By clicking the drop-down menu, we get to select a cancer type of preference, and the system will provide the CNV plot of this specific cancer type where all CNV frequencies are depicted. By examining the graph, we can focus on a chromosome of interest, for example, choosing chromosome 8 that presents increased variation frequencies, and the system will bring relative information from the PubMed bibliography that matches our chosen chromosome cancer type pair. This kind of information originates from triples that were extracted from paper abstracts with the help of an INODE triple extraction tool that employs both rule-based and ML-based techniques. The results are tagged with a ranking score that resembles the strength of this chromosome's association with the cancer. A record with a high ranking is proposed to be investigated more thoroughly by looking at the source paper it comes from, while a low-ranked record could be an indicator of a low correlation between the chromosome and the cancer. In the second tab, we can explore cancer cell lines that have been mapped to Cellosaurus, a knowledge resource on cell lines. By choosing a cell line from the list, we get genes, cytogenetic bands, variants, and neoplastic processes data related to this specific cell line. For instance, let's select the 5-8F cell line and check what we can explore from this as a starting point. With 24 results brought by the system regarding associated genes, in this demonstration we pick the highest ranked one and examine what kind of connections this gene might have with the cell line. The same procedure also applies to the rest entities, and thus we can look for interesting information to other fields as well. In a nutshell, the Cell Lines tool manages to combine APIs hosted by the University of Zurich and tools developed inside the INODE ecosystem, and offers the opportunity to a domain expert of exploring cancer cell lines in many ways and extract some more fruitful results.
Michael, I'd like you to tell me about your first impressions about the cell line tool that is currently being developed by the INODE team.
I have to say that I was pretty impressed by how fast we got to during just prototyping with limited data set and so on, to some usable data, which greatly showed also the power of combining our structural annotation, structured annotations, where we have defined entities, ontology codes, and so on, for lots of associated with our genome data, and to use these plus other information like positional information derived from our genome profiles to feed into the INODE system and retrieve meaningful information enrichment, especially directly leading to articles which would be useful for any user interacting in the way we envision by going to our website and using this as a jump-off point and into an information system. I expect that with the final version, this will be very useful and will deliver an additional layer, important additional functional layer to our data resource, which, as I mentioned before, is more of a static data content representing very nicely this type of genomic variations we are collecting and annotating and so on, but now really making this as then our data knowledge system. And
yes, thank you again. And so far, all the discussions that we have previously had, it seemed that the cell line tool would be easily and effortlessly embedded in your current structure, and you can host it without any concerns.
Right, so that's from what I see, and this is also facilitated by basically a kind of modern structure. The two systems have basically they are API driven, have open data exchange, so it's basically everything happens on a layer of data exchange and discovery APIs. We use an API compatible or derived from the Global Alliance for Genomics and Health Beacon system, which we co-develop actually, and integrating this with the INODE knowledge system is something which seems to me so far very easy and very smoothly going. Yeah, but that also depends on the nice support we have from the development team there.